Okay, so 11.3 is the kidney and osmoregulation. Well, in fact, you're going to study the excretory system in this chapter. So since the title is the kidney and osmoregulation, Ivy wants to start by discussing the two types of organisms. We have osmoconformer and osmoregulator. So we humans are osmoregulator, and other organisms are classified as osmoconformer. So just from the word itself, you can guess the meaning. Osmoregulator means we can regulate the osmolarity in the kidney. We can regulate the osmolarity in the blood, regardless of the environment. It stays the same. So the idea is homeostasis. The osmolarity of the blood is the same regardless of the environment. How about osmoconformer? Conform means to copy. Osmoconformers copies the osmolarity of the environment. So examples of your osmoconformers are your sea urchin, your starfish, your snails, your jellyfish, some species of sharks are osmoconformer. And you might ask why these aquatic organisms conform to their environment. Well, common sense will tell you that, of course, the osmolarity of the ocean changes depending on the place. And these organisms, if they don't change, if they don't conform to the osmolarity of their environment, the environment is hypertonic, then their cells will hypertonic shrink. The environment is hypotonic to the solution of their cell, then their cells will burst. So to prevent that, they need to conform to the osmolarity of the environment. Okay, but humans, we have we are capable of regulating our osmolarity because we have an organ called the kidney, and some of the other some aquatic organisms don't have a kidney to regulate the osmolarity in the fluids of their body. So what do we need to learn here in the excretory system? First of all, you need to know the excretory system of insects. That's kind of short. And then the excretory of humans, so our kidney, our excretory system, and the different types of waste product of the excretory system. Let's start with your insects. So insects have what you call a Malpighian tubule system, and they have a special way to excrete their waste product. Okay, so let me use the diagram picture used by our textbook. So this is your insect, and this is your digestive tract, this red one. It can be divided in parts such as your midgut and your hindgut. So mid meaning middle, hind meaning behind, so the back part of their digestive tract. Midgut, then hindgut. So in their digestive tract, of course, that's where they digest their food. But somewhere here in the midgut, you have tubules coming out of the tube, and these are called Malpighian tubules. These Malpighian tubules, that's where your excretory waste products enter. So basically, their food and their product of excretion mix here in the midgut. So what happens is, first you have active pumps for sodium, so sodium is pumped in. If sodium is pumped in, chloride, Cl minus, will follow, and through osmosis, water also follow. So together with the waste products such as your uric acid, now here it's in the gut, the digestive tract. Somewhere in the hindgut, your sodium goes back into the hemolymph, or their fluid, the fluid of the body, such like their blood. So sodium goes back, potassium goes back. Obviously, through osmosis, water will also go back. However, uric acid, or the waste product, stays here inside. So they excrete that. Uric acid is excreted together with the waste product of their digestion. So digestive system, and they have Malpighian tubules connected to the digestive system. Their waste product from excretion is mixed with the waste product also of their digestion, and then it comes out as a semi-solid waste called uric acid. Okay. Now let's talk about the excretory system of humans. So we have an organ called the kidney, and you need to know the structure and function of the kidney and its functional units called the nephrons. I have a drawing here. Hold on, please. Look at our organ for osmoregulation. The kidney is made up of functional units called nephrons, and this is how a nephron would look like. It's basically a tube that filters the blood, and it will have different parts: your Bowman's capsule, your proximal convoluted tubule, loop of Henle, distal convoluted tubule, collecting duct, and then this is going to go out of your kidney as your ureters, and your waste product will be stored in your bladder and then out of the body through your urethra. What you need to know for IB is to know what happens here, 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 and here. So the physiology of the different parts. So the first one you need to know is blood composition. So right here, these are blood vessels entering the kidney. So you call this the renal artery that becomes smaller and smaller, or capillaries called renal arterioles. And then this specialized bed of capillaries, we call it glomerulus. So they're still capillaries, but it has a specific name, glomerulus. And after filtration, it will become your blood vessel called your renal venule, which will become your renal vein. So then again, renal artery becoming your renal arteriole, and then your glomerulus, and then out as your renal venule or your renal vein. So first of all, you need to know what is the composition of blood. Comparing the composition of blood in the renal artery here and then in the renal vein. So maybe you can have a listing here. So in the renal artery, obviously you're going to have toxins. I think we've listed it. Please check your notebook. We have toxins. We have oxygen here. We have your excretory waste products here. There's water, and there's also salt. Okay, and then your blood will be filtered. Obviously, after filtering, there's more of your carbon dioxide because the kidney cells underwent cellular respiration. So you have more of your carbon dioxide. No more toxins. It was filtered out. No more excretory waste product. It was filtered out, but still with water and still with salt. Okay. Next, we have to look at what's happening here in this part. So as you can see, the glomerulus is surrounded by the first tube of the nephron. You call it the Bowman's capsule. And here we have a process called ultrafiltration. So your blood is super filtered before it can, before the waste product can go here in your Bowman's capsule. Zoom in that portion of the glomerulus in your Bowman's capsule. So this was your artery, this was your glomerulus, and then your renal venule, renal vein. If you can see, if I zoom that in, from the capillary from the glomerulus to the Bowman's capsule, it has not just one, not just two, but three layers where blood needs to pass through before it can get into the Bowman's capsule. First of all, the endothelium of the capillaries have fenestrations, those holes. So everything else can pass through except your red blood cells. They're too big to pass through the capillary endothelium. Next layer is your basement membrane, your glomerular basement membrane. This basement membrane has glycoproteins. So proteins cannot pass through here because proteins cannot pass through glycoprotein layer. Okay. Then lastly, you have your podocytes or your foot processes. They imagine them as feet pushing against the wall, trying to prevent things from filtering out. So three layers: the endothelium, red blood cells can pass through that. Basement membrane, proteins can't pass through that. And then your podocytes, so it prevents smaller molecules from being filtered out of the blood. And then the rest of the molecules can get into this part called the Bowman's capsule. So we need to know what's happening here in the proximal convoluted tubule. After you filter the blood, so what's going to happen to those filtrate or the liquid that you filtered out from the blood? What's going to happen here in the proximal convoluted tubule? To make it simple, most of the molecules that were filtered out will go back into your the tissue of the kidney. It will go back to the body to leave the tubules here in the proximal convoluted tubule. The word for that is reabsorbed. So what will be reabsorbed here in the proximal convoluted tubule? So this is your proximal convoluted tubule cross section. We're looking at top view of the tube. So this is the inside of the tube. This is the outside of your tubule. So what you need to explain here is the method of reabsorption in the proximal convoluted tubule. So reabsorption means simply means for the absorption back into the blood or back into the tissue of your kidney. So how are the filtrate here reabsorbed or moved back into the kidney, the tissue of the kidney, or back into the blood vessels in the tissue of the kidney? So first of all, you need to know the reabsorption of sodium ions. So if you look at this one cell right here, so I have a drawing here of this cell, and you go like, wait, miss, that looks like the cells of the small intestine. So yes, the structure is similar, although it's not as columnar. This is a cuboidal cell, but the same thing here. It has a brush border. It has microvilli. Okay, anyway, so this is something you've learned in the small intestine already, so very easy for you to remember. So how is sodium moved or reabsorbed from the inside to the outside here? So first, well, you have pumps here, sodium, potassium pumps. So this sodium is pumped into the blood, okay? And this creates a concentration gradient that allows movement of sodium from the inside to the cell. At the same time, glucose is co-transported together with sodium. Then once inside your cell, the glucose will pass through a protein channel, a glucose channel, into your blood. Okay, so how does reabsorption of sodium and glucose happen? Well, sodium is pumped into the blood and glucose is co-transported together with the sodium, and through the glucose channels, it goes back into your blood. You also have to know how chloride ions is reabsorbed into the blood. Well, chloride is Cl minus, right? So if sodium goes out, that's positive, Cl minus will just follow sodium passively. So Cl minus will diffuse passively into your blood. And if you have your sodium, if you have your Cl minus, if you have your glucose, that sets up a concentration gradient that will cause water to move from inside to the outside through osmosis. Okay, so again, sodium is pumped into the blood, glucose is co-transported and it moves into the blood. Chloride ions, that's Cl minus, right? It will follow the movement of the positive ion. So it will just simply diffuse into the blood. And then if all of those ions are present here, then through osmosis, it will cause water to move from the inside of your tubule out, okay, into the blood.